Our next speaker is Mark Musen from Stanford University. Mark is going to speak about knowledge technology to accelerate open science in addressing the COVID-19 pandemic.
Thank you all. And again, thanks for the opportunity to be here. There's been a lot of discussion in the lay press about how the COVID-19 pandemic is going to change a lot of features of society. One element of society that actually hasn't been discussed a lot is that COVID-19 is going to change science and the way we do science in some profound ways. And what I want to talk about today is how some of the work we're doing at Stanford may be helping to push that change forward, particularly in the context of the current epidemic. And now I can't advance my slides. There we go. I'm sorry. The main thing I want to have you take away is that science in the past 10, 20 years has changed radically as people are recognizing the importance of making available online the very data that scientists are using to form their hypotheses and to publish their papers. That there's been a shift as we recognize that the output of science is important, not just in terms of publications, but in terms of basic data, so that other scientists can go and look at those data, can verify that the results of the experiments are correct, and also can take those data and reuse them and potentially make new discoveries. And nowhere has this been more important than the case of the COVID-19 epidemic, where suddenly all these data that are filling up resources all over the world with information about COVID-19 suddenly are available, and people want to be able to have access to them quickly, want to be able to use other people's data to make new observations. And there's just enormous excitement about the whole idea of open science and the opportunity to take advantage of data that already exist to be able to make new observations and hopefully to address the epidemic. That's all the good news. The bad news is that although we have lots of data out there, it's kind of like having a library where you don't have a good catalog, where you don't know where all the resources are, where you can't actually easily find what you need to find. And so the scientists who are most interested in being able to find other investigators' data and learn from them are kind of stymied. And they're stymied for a reason that's going to sound rather arcane. They're stymied because they don't have good metadata, the data that describe what the data sets are all about. Let me just show you why I say that. In this, first, let me just emphasize that the buzzword that has become so frequently used now to describe the problem is the idea of trying to make data that are fair, that are findable, so you can actually search for them, that they're accessible, that even once you find the data, you can actually download them and look at them, that they're interoperable, that you can take your data and mix them with my data, and we can actually get something better than the sum of the parts, and that the data are reusable so that they're explorable by others. So this buzzword of having data that are fair pervades biology right now. And the challenge, as I said, is that most data are not fair. So here are the metadata, or a tiny piece of the metadata that describes samples that are in a repository at the NIH called GEO. And these results show you in several records how scientists have described the age of the patient, from age to age to age to pop age to age after birth to age in years to God knows what. If you're a scientist and your goal is to find a specimen that's described in a repository where you want to look at the data that were accrued from some experiment, you have to know what to search for. And if you have to know, and if you're interested in patients of a certain age, you have to know how the investigator might have chosen to represent age in one of a million different ways, including a million different typos. We looked at a repository called Biosample, another NCI repository, and wanted to really quantify how bad are the metadata, which means how bad and how difficult is it to search for someone else's results. Well, 73% of the Boolean metadata values are not true or false. They're values like non-smoker or former smoker. 26% of the integers don't parse as numbers. And 68% of the metadata entries that are supposed to represent terms from standard vocabularies, well, are not terms from standard vocabularies. And what that means is if you're an investigator and you're trying to find a particular set of data, you're often really stuck because the metadata are terrible. And why is that important for COVID-19? Well, if we want to find relevant studies, if we want to be able to integrate new results with those that are already available, if we want to re-explore existing data sets, if we want to verify published claims, we may have a lot of difficulty finding the right answers. And one of the things that I find remarkable is that just in the past couple of weeks, there's been this absolute flurry of activity in the COVID-19 community demanding that we get data that are available and that are more fair, data that are more accessible, data that are more reusable. And the question becomes, how can we actually come up with better data like that when, frankly, the data that exists online are very difficult to search and very difficult to use? Well, like many other people in this session, we have technology that we're trying to repurpose specifically for COVID-19. We have technology we've created at Stanford called Cedar, where Cedar is a center for expanded data annotation and retrieval. And Cedar basically offers a three-step process where initially we author metadata templates. We decide for different classes of experiments, for different kinds of experiments in virology, in biochemistry, and whatever, what are the kinds of experiments that people do? And can we come up with a way of standardizing what the metadata for such an experiment should look like? We then have a mechanism within Cedar that allows us to annotate those templates. We basically fill in those templates with terms from controlled terminologies that are consistent with well-defined data types. And ultimately, that gives us the ability to take the metadata that we enter into Cedar and use those metadata to annotate the data sets that are stored in the repositories that investigators ultimately want to explore.
1 minute.
One of the things we also do is to look at those data so that we can easily use those data to make it easier for people to fill in the blanks of the form. So, for example, here's a form for filling in information about a specimen for a repository called Biosample, and the drop-down menu gives you all the controlled terms that might be appropriate for the tissue. However, if we want to look at what the disease might be, if we already said the tissue is lung, then because we know a lot, we can use AI and say, well, look, we don't need to provide all the possible values. We just look at lung cancer or chronic obstructive pulmonary disease or other kinds of conditions that might be associated with lung tissue. If we say the tissue is brain tissue, then our menus can be much more simple, and we can specify, for example, what are the kinds of diseases that might be associated with a tissue coming from brain. What that means is when we create metadata, we don't deal with the mess that we currently have, but rather we have metadata that are really tailored for the situation and that use the kinds of terms that people want to search for. In the past couple of weeks, the Go Fair organization, the people in Europe who are promoting the FAIR principles in the strongest way, have created something called the Virus Outbreak Data Network, or VODAN. And one of the things that VODAN is doing is using technology such as Cedar in order to be able to make it easier for investigators to create data related to the epidemic, data related to virology, data related to all the kinds of experiments that people need to do in this area, and make this data more readily available. We know that data are never going to be fair unless we have common templates, when we have controlled terms, and we have technology that makes this all easier. And so what gets us very excited here at Stanford is the ability to take our existing technology known as Cedar and to repurpose it for the purpose of making it easier and faster and more reliable for workers in the COVID area to be able to get the kinds of data sets they need in order to do the kinds of analyses that they view it to be most important.
Thank you very much, Mark. And I'm just struck in this session by how many people have pivoted their organization and their efforts to be relevant and useful for COVID. It's quite inspiring.